Hi there. This is part two of major events in evolution. In this part, I'll talk about some additional important events, including events in the evolution of molecules, cells, organelles, and entire organisms. In part one, I talked about where some of these molecules came from, some of the biological molecules, including water, CO2, methane, ammonia, amino acids, peptides, etc. And then there was this period of about 250 to 500 million years when there was an evolution of some of these that were then used in the first organisms, which were RNA-based. So I want to talk a bit about RNA first. While DNA-based organisms probably didn't originate until about 3.5 to 3.8 billion years ago. Previous to that, RNA-based organisms evolved, which were probably the first organisms on Earth. And that included the development of ribozymes, and one of the largest ribozymes is the ribosome, ribosomal RNAs. RNAs form many different structures. They're almost always single-stranded, but then they fold up into characteristic shapes, including shapes that make them catalysts or ribozymes. One class are introns, and we'll talk about those later in some of these videos. But here's a group 1 intron up here, and another group 1 intron here, where they're bringing the two exons together and splicing those together. And that's a function that's performed at least in group 1s and group 2 introns, where they're self-splicing. They don't require any proteins for the splicing reactions. So RNA and ribozymes evolved or started to form very early on Earth, probably 4.1 to 4.2 billion years ago, and became part of the both the enzymatic and the hereditary or genetic material of the initial organisms. One of the largest and probably one of the most complex ribozymes are ribosomes, and it's the large subunit that has the catalytic activity of the ribosome. And so its function is to take an mRNA, and by using tRNAs to essentially decode a message, it will build peptide bonds between adjacent amino acids that are being carried by the tRNAs. And these are found in all membrane-bound life on Earth. So they've been there since the beginning. They were very successful in building proteins with high fidelity. And so that's one reason why they were selected for very early on Earth, probably 4 billion years ago or more. All cellular life on Earth have ribosomes. That includes bacteria, archaea, and eukaryotes. However, the ones in bacteria and archaea are a little smaller than those in eukaryotes. They have smaller ribosomal RNAs, and they have a smaller number of proteins, so their molecular weights are also a little smaller. Actually, archaeal ones are slightly larger than bacterial ribosomes, and are a little more complex. And the process of translation, that is, taking an mRNA and translating it into a protein, requires several different RNAs, including the mRNA, which is carrying essentially the message only. And then the ribosomal RNA, the small subunit RNA, first attaches to the mRNA, and then that joins with the large subunit ribosomal RNA in the ribosome, and then tRNAs deliver the amino acids to the ribosome to decipher the message and add the amino acids together as a polypeptide, which is eventually, the protein. So those that are 50 amino acids or shorter are considered as peptides, and those that have more than 50 amino acids are called polypeptides or proteins. So early on in evolution, between let's say 3.8 and 4.2 billion years ago, RNAs performed many functions in the cells, those initial cells, including enzymatic functions and handling the inheritance or genetic functions. Later on, some of the RNA functions were taken on by peptides and later by polypeptides. But many of the RNA functions remain even today, including the ribosome, which is a large ribozyme, and other things like NADH, ATP. Those are RNA molecules that still are functional as RNA molecules. So at the beginning, they were not nearly as complex. So this shows an evolution of RNAs involved in translation. So in this phase one, that is between 3.8 and probably 4.2 billion years ago, the tRNA was very simple, mRNA was very simple, and the small subunit and large subunits were also very simple, although they already had the function of joining amino acids together with peptide bonds. These two types of molecules above, class 1 and class 2 aminoacyl tRNA synthetases, these are the ones that attach the amino acid to the tRNA. Now, initially, back 3.8 to 4.2 billion years ago, they were very simple and probably consisted mainly of RNA. By phase 2 here, 3.6 to 3.8 billion years ago, they were already peptides and proteins that had taken over the job of the initial RNA. So these ones up here from very early stages became proteins rather than RNAs, whereas these down here, the ribosomal RNAs, mRNAs, and tRNAs remained as functional RNAs, but they tended to get larger and larger and larger and more complex as time went on. And if you look at the flow of genetic information in cells, that is the central dogma, DNA is replicated here, it's transcribed into RNA, and then mRNA is translated into protein. But at the very beginning, the first biological molecules were RNAs only, and maybe a few peptides. But there were no proteins and there was no DNA. So this whole process started in the middle here with the RNA performing both the inheritance or genetic portion of carrying on the instructions for making peptides and RNAs, and the RNA functions as well, the enzymatic functions. Not long after, and you can see this in those aminoacyl tRNA synthetases, peptides and proteins became important because they were a little more stable than the RNAs, so that they could perform those functions for a longer period of time before they degraded. And so now we have a system probably 3.8 to 4 billion years ago where the organisms consist of RNAs, a bunch of different RNAs, and proteins as well. And already by that time, the proteins were being translated using ribosomes. However, there was a non-ribosomal way to make peptides, small peptides, and it's actually used still today, a non-ribosomal protein and peptide synthesis system. But it's not used as much as ribosomes are, because ribosomes are much more reliable and they have a higher fidelity of making those proteins, so they can make longer and longer proteins. And then sometime later, probably around 3.5 to 3.8 billion years ago, a relative of RNA, a 2'-deoxyribonucleic acid, DNA, began being used instead of RNA because it could form double helices and protect the DNA. And also then you would have essentially two copies from which to replicate. the nucleic acids, the DNA. So it's much more stable, and it can be replicated with higher fidelity. This allowed genomes to get larger, and therefore the cells could become more complex because they could carry more genes. So far then, in this tree of life, there's the bacteria, archaea, and eukaryotes. We're about here on this little root from 4.2 to about 3.8 or 3.5 billion years. And that's where RNA first became established, peptides and proteins became established, and then ultimately by this time, the organisms that survived, at least, were selected for, were based on DNA. So I want to talk now about a few other events. I already talked about asteroid impacts, so we've dealt with that a little bit. And then I briefly touched on increases in oxygen and photosynthesis, a bit about multicellularity, and then how eukaryotes got their nucleus, and then mitochondria, how those were acquired and led to eukaryotes having oxidative phosphorylation capabilities. As I stated in a previous video, and will probably remind you in many other videos, the basic phylogenetic tree here with the bacteria over here and archaea and eukaryotes also has these lines coming across to where a bacterium entered an archaeal cell to become the first eukaryotes with nuclei, and then another bacterial cell came over that had oxidative phosphorylation capabilities and became the mitochondrion. And then another bacterium from the cyanobacterium came over and became the plastids or chloroplasts. And then we have other events too. So you have these organisms joining in endosymbiotic relationships and eventually some becoming organelles. So you can look at the tree of life as being a network rather than just a simple tree. So if we go out of this 3.8 to 4.2 million year old lineage here, and we have the bacteria and archaea coming out of that. You see the eukaryotes don't come out of that line, but they become entwined in different areas. The bacteria and the archaea become entwined here and join as a network, because initially the nucleus became part of these archaeal cells to become the eukaryotes, and then a mitochondrion, and then a chloroplast, and other organisms have joined with other eukaryotes here. So it's a eukaryote in a eukaryote, but that was really a bacteria in an archaea, etc. So you don't have a simple tree, but you have a network, which is the tree of life. Or if you want to say, it's the network of life rather than the tree of life. And it turns out it's even more complex than that. So not only do you have these major endosymbiotic events leading to new organisms with other organelles, but there are minor movements of DNA and other endosymbiotic events that are occurring on each one of those lines. So then evolution really, instead of a tree, is really a netted pattern rather than a simple bifurcating tree. And even if you look on these seemingly straight branches, those branches consist of populations of interacting organisms, genetically interacting organisms, either trading DNA or mating, coming together to join DNA, or becoming endosymbionts of one sort or another. So you have a net within a net in this case. And again, going down to the bottom of this figure, note that there are only two branches coming out of here, the eubacteria and the archaea, and really the eukaryotes are produced by joining of a member of each one of those. And here's another way of looking at that. Originally, people used to think this, where there was this first eukaryotic common ancestor, and you have bacteria coming out over here, archaea coming out of here, and then the eukaryotic line coming out of that. Eventually you get a mitochondrion from an alpha proteobacterium coming over and becoming the mitochondrion, and then you can have another one, the plant, coming out at a later time, the cyanobacterium becoming the chloroplasts. However, a much more accurate model is this one, where you have this last universal common ancestor, which diverged into bacteria and archaea, and then a bacterium and an archaea came together to form those first eukaryotes, first containing a nucleus, and then containing a nucleus and a mitochondrion, and then containing a nucleus, mitochondrion, and plastid in the case of plants. And then there were other events too to form other organelles in a similar fashion by endosymbiotic events between a eukaryote and another eukaryote. So Darwin was essentially correct in that you have these bifurcating trees, but what he didn't know was that the trees interact with one another so that you can get DNAs or whole genomes that are moving from one organism into another organism and affecting its evolution. So some things that have driven endosymbioses and mutualism, essentially, in evolution, one is protection. Organisms can protect one another. Second is motility. So some cells which aren't motile can actually be helped by other organisms that are motile by helping to move them around. And this actually is quite common. Third one is carbon, energy, and nutrient acquisition. So this could be to share resources. If one organism can form certain things, the other one can form other parts. They can actually complement one another. They can also this can be driven by community cooperation and mutualism, which is part of the number 3 as well. It can help them to adapt to new habitats, such as acquisition of a mitochondrion to allow those organisms to grow in high regions of higher oxygen concentrations. And finally, 6, the symbiotic relationship can change an organism as far as where it can live, or its structure, or where it can survive and adapt. So here's the general tree of life with bacteria down here, archaea, the root would be somewhere in here, and then the protists here, the excavata, and then now this is Rhizaria chromista alveolata, but now they've changed it to the SAR group, Stromatophiles, alveolata, and Rhizaria. And then the plants here, amoeba, fungi, and animals. If evolution were a simple tree, then it would make things a little easier, but it's not just a simple tree, as I've indicated already. So in order to form eukaryotes, an archaeal cell, which came off of this branch, joined with probably a planktomycete to form a cell now, a more complex cell, a more complex archaeal cell with a nucleus. So here's a simple view of that. Here's an archaeal cell, and it differs from a bacterial cell in several aspects, and I'll be going into more detail about that in a subsequent video. But planktomycetes have internal organelles, including some that have a nucleus-like organelle. So if those join together, now you have a eukaryote that is formerly archaeal cell with a nucleus now. And just to show you one of these planktomycetes, this is Gemeta obscurula. You can see here, there's the cell membrane on the outside, the cytosol here, and then there's an internal membrane-bound structure. It has a double membrane, just like a nucleus. And then it has structures also that look like nuclear pores. And so this one, here's another view of that with the cell membrane and then this nucleus-like structure. And here's a slightly closer view of that. There's the cell membrane and the intracellular membrane, and then a double membrane structure here that looks like a nuclear envelope. And it has the DNA inside of that nucleus-like structure. These other dark structures over here are ribosomes, which would appear inside nuclei as well as out in the cytosol. And this is just another way to look at the same thing. This is a freeze fracture view in an electron microscope, and you can see these vesicles in there as well as the cell membrane out here, and then this double membrane structure here, which is the nuclear envelope, and then the what's being called a nucleus-like structure. To me, it looks like a nucleus. So if someone says bacteria have no nuclei, I would say that's not necessarily accurate. So to build up a eukaryotic cell then, you have a progenote cell, which is an archaea, joining with a planktomycete cell, and that produces the first nucleus, which is essentially an endosymbiont, which is a planktomycete inside of an archaeal cell, or it could be vice versa also. And we see that this eukaryotic cell here has characteristics of both the bacterium and the archaea, which is true. And in a more detailed tree, so these are the bacteria over here, and then down here are the archaea, and this green line coming down here, those are the eukaryotes, including us. And you see that they're very closely allied to archaea, indicating that the eukaryotes definitely did arise from one branch of the archaea, but they picked up some bacterial genes as well in the form of planktomycete genes, mitochondrial genes, and in plants, cyanobacterial genes as well. You can see this also if you look at the gene characteristics and protein characteristics of each of the types of cells. So everything in blue here, the number of genes, transport of inorganic ions, ribosomal operons, etc. All these processes are bacterial-like, and archaea have some of those, especially since these are all unicellular organisms, then cell division and things like that would tend to be similar. However, when you look at ribosomal proteins, you look at translation among those groups of organisms, as well as RNA polymerases, DNA replication, etc., then eukaryotes and archaea have more in common than they do with bacteria, and that's also indicated by that phylogram I just showed. While a few types of eukaryotes have no mitochondria, so they cannot perform oxidative phosphorylation, a large number of eukaryotes have oxidative phosphorylation that comes from mitochondrial functions. And that came out of the alpha proteobacterial line entering one of the eukaryotic cells, first to be sort of an mutualistic endosymbiotic, and then eventually the mitochondrion, the alpha proteobacterium became the mitochondrion. And this occurred somewhere around 2.2 to 2.4 billion years ago. And that was at a point when oxygenic photosynthesis in cyanobacteria had begun. So they began pumping out oxygen into the atmosphere by breaking water, essentially, to get electrons and protons to make ATP. And that increased the amount of oxygen in the atmosphere, but that was toxic to most anaerobes. And so at that time, the anaerobes either went deeper in the water to make sure that they stayed away from the oxygen, or they died, or they formed some sort of relationship with an organism like an alpha proteobacterium that could handle the oxygen. We know that the amount of oxygen was increasing in the atmosphere because of this banded iron, the ages of these banded iron layers in the rocks that were at the bottom of the ocean and the bottoms of the lakes. What is occurring is when iron is reduced, the reduced forms of iron are very soluble in water, so they would be soluble in the ocean, soluble in the lakes, etc. But when you have oxidized iron in the form of rust, essentially, it's not soluble. So as soon as that starts reacting, the iron oxide starts precipitating out of the water and forms these rusty layers on the ocean floors and the lake floors. So the atmosphere essentially went from being a primarily reducing atmosphere to an oxidizing atmosphere. And that creates big problems for anaerobic microbes. They die unless they team up with an organism that can handle the oxygen in some way, so that their contents are essentially not oxidized. So at that time, all life was in the water because it was being protected by the water from the UV radiation. But the concentration of oxygen was very low in the water, so anaerobes flourished. That included bacterial anaerobes as well as the initial eukaryotic anaerobic organisms that at that time only had a nucleus. The atmosphere also had very low oxygen, so that there was no oxygen in the atmosphere moving into the water either. So here you have bacteria that grow in low oxygen and eukaryotes that grow in low oxygen as well at that time. Then cyanobacteria start churning out oxygen that goes into the atmosphere and moves worldwide. And so some organisms start to change, start to mutate so that they can grow in high oxygen. And probably initially they were just sequestering the oxygen using some molecular mechanism. But later some developed a way to use that oxygen to join it with hydrogen, which was plentiful, and to siphon off the energy from that reaction. It generates a lot of energy to make ATP. So the next step was for one of those eukaryotic cells to collaborate with one of the bacterial cells that could do oxidative phosphorylation, and to take that on as a new, first an endosymbiont, and then to convert that into an organelle, which became the mitochondrion. So here's the next step in the evolution of those eukaryotes. There's the primary eukaryotic cell joining with an alpha proteobacterium, also called purple bacteria, to form that endosymbiotic relationship that then could survive the higher levels of oxygen and move into new niches in the environment. And that became the mitochondrial eukaryote then. So looking at the tree again, we have a nucleus from a planktomycete moving into an archaeal cell, and then an alpha proteobacterium entering one of those cells to become a mitochondrial eukaryote. And then to form plants, there was another endosymbiotic event between a cyanobacterium and a mitochondrial eukaryote to become the first plants, which were red algae and glycophytes, and then eventually green algae as well. So then that would convert a heterotrophic eukaryote into a photosynthetic autotrophic eukaryote. the plants. So this became an advantage for those organisms because they no longer had to essentially search for their food, but they could just hang out in the sunlight and produce their own food, their own carbon compounds. So here's that process to make a plant cell then. You take a mitochondrial eukaryote, join it with a cyanobacterium, which initially was a mutualistic relationship, then became an endosymbiotic relationship, and finally the cyanobacterium was converted into an organelle. There actually was a second such event to become the rhizaria here, where in some rhizaria, another cyanobacterium at a different time, a later time, entered a rhizarial cell or a non-photosynthetic protozoan cell to become a rhizarian, and now has what's called a chromatophore inside of it. It's not called a chloroplast, but essentially it's analogous to a chloroplast, but it was two separate independent events. So here's this tree of life again, where we have the overall phylogenetic tree with these different branches or these different organisms joining together in a mutualistic and then an endosymbiotic, and then ultimately an organelle, becoming an organelle. And these are not the only ones. There's a lot of other endosymbiotic events that have occurred, as well as what are called horizontal gene transfers of one gene at a time or 10 genes at a time. So it's happened a great deal in evolution and has caused many different types of cells to arise. So it's been a big impactor in evolution. And if you think about it, you're joining one genome with another genome, or two genomes, or three genomes. So those all have to interact in a coordinated way. And in evolution, they have essentially, although there are problems with joining so many genes together and their protein products, joining those together to make sure that they're compatible. So it doesn't always work, and probably has happened many times where it did not work. But in many cases it has worked quite well to form, the mitochondrial eukaryotes and photosynthetic eukaryotes and other types of eukaryotes as well. It has also happened in prokaryotes also, and I'll talk about that in a future video as well. Okay, that's the end of part 2 of major events in evolution. I'll extend on some of these in the next video, which will be the final part of this, part 3, in major events in evolution. So thanks for listening and watching, and I will talk to you later.